Hello. Good evening, all. Am I audible?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay, thank you. So I'll start sharing the screen and we will begin with our session. So I'll start sharing the screen. So people are still joining in. So I hope from the next session, like everybody gets joined by 6:05. So it would be much better. So else I need to come back here and admit, which won't be feasible for me. So I hope you guys join a little early from the next session. So now I'll start sharing the screen. Can anyone confirm whether my slides are visible or not, and whether they are clear or not?
Ma'am, slides are visible, but they are blurred in image.
Okay. Just wait a second. Is it any better now? I think they are still blur, right? Just
Yes, ma'am. Yes, ma'am.
is it better now?
Yes, ma'am.
Okay. Uh, okay, we'll start with our session. So, I welcome you all to our first problem solving session. So, the course name is Molecular Biology, and this course is being taken by Professor Vishal Trivedi from Department of Biosciences and Bioengineering from IIT Guwahati. So, in this first week problem solving session, we will be covering week 1 lectures that has been already covered by the Professor Vishal. So the first lecture is cellular structure of prokaryotic cells. The lecture second and third, they consist of cellular structure of eukaryotic cells, and the lecture 4 and 5, they consist of cell fractionation part 1 and the part 2. So I'll briefly introduce myself as well. So I am Pooja Yadav. I am a PhD scholar and a PMRF fellow in Department of Biosciences and Bioengineering from IIT Bombay, and I am a PhD student in molecular virology lab. So we will begin with our session. So first we'll go through the question, and I expect you to try to answer those questions first. Then we will look into the concept of for those questions. But first, please try to answer the question by yourself. Then we will discuss the answer. I hope this is clear. So shall we begin with our questions?
Yes, ma'am.
Okay. So question number first, it states that chemotaxis is defined as first, a movement of cell directed by chemical gradient. Second, movement of cell in response to light. Third, movement of cells driven by intrinsic factors. Fourth, cell movement in response to changing temperature. Uh, Please try to answer the question. Either you can unmute yourself and answer, or you can also write in the chat box. For those who are joining late, please try to join by 6:05, so I don't need to come back and then give you the permission.
The movement of cell directed by chemical gradient.
Okay, we have one answer, and in the chat box as well, we have A as an option A as in for as our answer. So the correct answer is the movement of cell directed by chemical gradient. So let's look into what is chemotaxis. So basically the chemotaxis, it refers to the movement of cell or an organism. Here you can see a bacterial cell in response to chemical stimuli. So in the presence of environment, there are certain chemical stimuli like nutrient for bacteria are present. So in the presence of those chemical stimuli, the bacteria move either towards it or away from it. When a cell, it moves towards the chemical gradient, it is a positive chemotaxis, and when it moves away from the chemical gradient, it is negative chemotaxis. So chemotaxis may be either informed foraging or it is a cue based navigation. In case of informed foraging, what happens is a bacteria or a cell.
Excuse me, ma'am.
Yeah.
Suddenly your screen is being stopped.
Okay, so uh, you cannot see my slide.
Ma'am, it's pretty unclear and suddenly we cannot see your frame moving.
Okay. So I think there is a connectivity issue. I apologize for that. So let's try if it can get any better. Stop sharing screen. Are you guys able to see the slides or not? I think my slides are still not moving. I hope you guys are able to see the slides now clearly.
Yes, ma'am.
Okay. Sorry about today's connectivity. We'll figure it out in the next class. Please bear with me today. Okay, so let's begin with the what is chemotaxis. So chemotaxis, it refers to the movement of cell or an organism. Here we are talking about a bacterial cell in response to a chemical stimuli. Uh, it could be nutrient for a bacteria. So when a cell moves towards the chemical stimuli, it is positive chemotaxis. When it moves away from the chemical stimuli, it is a negative chemotaxis. So the chemotaxis, it can be informed foraging or it can be cue based navigation. So in informed foraging, what happens is chemotaxis, where a nutrient is an attractant, the bacteria is moving directly towards the nutrient, while in a cue based navigation there is a mediator to which a bacteria move, then it leads to the main nutrient or another chemical. So where we can see all these chemotaxis, it can either be in gastrointestinal tract. The bacteria present in the gastrointestinal tract, they are chemotactively active towards various chemical stimuli. The bacteria present in soils may be attracted to the chemical present in the soil, which is leading to the roots of the plant, or they can also be found in the aquatic environments where multiple chemicals are present where the bacteria are attracted towards them. So the correct option for this question would be movement of cell directed by chemical gradient. So if you guys have any doubt in any question, you guys can unmute yourself and ask, or either you can write in the chat box as well. So I hope question number 1 is clear. Okay, let's move to our question number 2. Question number 2, it states that peptidoglycan is polymer of option 1 NAM and NAG, option 2 chondroitin sulfate and heparin sulfate, 3 agarose and pectin, 4 glucose and galactose. Please try to answer this question. We have option 1 as
Option A.
okay. Is there any other option that might be correct for this question apart from option A? The correct option for this question is indeed option number A, NAM and NAG. Let's look into the concept of for this question. So what is peptidoglycan? So peptidoglycan is basically a part of cell wall of bacteria. So there are basically two kind of bacteria, gram positive bacteria and gram negative bacteria. So this differentiation is because the gram positive bacteria can take up the gram stain and they show the purple color in the gram staining, while the gram negative bacteria, they cannot take up the gram stain, so they remain colorless, or we need to add a different color dye to differentiate among gram positive and a gram negative bacteria. So in gram positive bacteria, the thickness of peptidoglycan is comparatively thicker when we compare to the gram negative bacteria. And what is this peptidoglycan composed of? It is basically a polymer of NAG, N-acetyl glucosamine, and N-acetyl muramic acid. it is linked by 1 beta 1 4 glycosidic bond with a short peptide chain attached to NAM subunit. These peptide chain are typically of L-alanine or D-glutamic acid or L-lysine or D-alanine. So here you can see these peptidoglycan, they are composed of alternative subunit of NAG and NAM, and these NAG and NAM, they are joined by 1,4 beta 1,4 glycosidic bonds, and these NAM units are connected by short peptide chain of L-alanine and D-glutamic acid or L-lysine and D-alanine. So what is the function of peptidoglycan in the cell wall of bacteria? They provide the structural integrity to this bacteria. It maintains the cell shape and it prevents from bursting of the cell due to the osmotic pressure. It provides the protection against environmental stress and mechanical damage, and they can be antibiotic target. It is a primary target for many antibiotics like penicillin, which inhibits the enzyme involved in its synthesis. So, the correct option for this question is option number 1, NAM and NAG. I hope this question is clear. Okay, let's move to our next question. Question number 3. The main reason that gram-negative bacteria lose their crystal violet dye during the decolorizing step is: first, they have a thicker peptidoglycan layer that traps the dye. Second, their cell wall is more resistant to dehydration. Third, they have an outer membrane containing lipopolysaccharide that are dissolved by the decolorizer. Fourth, the crystal violet dye is unable to penetrate the cell wall. Please take a few minutes and try to answer this question.
Ma'am, option C, ma'am.
Okay, we have two answer, option A and option C, in the chat box. Is there any other option correct from option C or A? So the correct option is, so we have one more option, which is 4. The crystal violet dye is unable to penetrate the cell wall. Okay. So the correct option for this question is option number 3. They have an outer membrane containing lipopolysaccharide that are dissolved by decolorizer. So here you can see in this question we have differentiated between the cell wall of a gram-negative bacteria and a gram-positive bacteria. So gram-negative bacteria, they has thinner peptidoglycan layer and it has a outer membrane of lipopolysaccharide. While in a gram-positive bacteria, we have thicker peptidoglycan cell wall and it is covered by lipoticoic acid. So when we do the gram staining, first in the gram staining, what we do is we add crystal violet dye to the bacteria and iodine, which initiate the take up of dye into the bacterial cell. So since the peptidoglycan of since the so dye gets inside into both gram negative and a gram positive bacteria, then we add alcohol, so it function as decolorizing of the bacterial the dye in the bacteria, and the peptidoglycan of gram positive bacteria is thicker, so the dye cannot come out. While in case of gram negative bacteria, the peptidoglycan layer is thinner, so while adding the alcohol, these dye they get washed off and they become colorless. So while the gram positive bacteria, it shows violet color in the gram staining when we do the gram staining, while the gram negative bacteria, it does not give any color. So to differentiate whether gram positive bacteria are present or not, we add a different dye, which is a safranin dye, which give the red color to the gram negative bacteria. So since the gram negative bacteria has a thinner peptidoglycan, as well as the outer membrane of the gram negative bacteria is of lipopolysaccharide, which is easily dissolved in presence of alcohol or a decolorizer, so it does not take up the gram staining, it loses the gram staining. So the correct option would be they have option number 3. They have an outer membrane containing lipopolysaccharide that are dissolved by the decolorizer. I hope this question is clear. So if this question is clear, let's move to our next question. Question number 4th. The function of the inner membrane of the nuclear envelope is: first, structural support; second, mRNA synthesis; third, DNA replication initiation; fourth, regulation of nuclear pore. Please try to answer this question. We have one option, which is option number 2 in the chat box. Is there any other option which is correct for this question apart from option number 2? In this question, the function of inner membrane of the nuclear envelope has been asked. Please try to remember the structure of nucleus and the nuclear envelope, and again try to answer this question. Please try to imagine the structure of the nucleus and again try to answer the question. We have two options apart from option number 2, option number 4th and option number 1st. Option number 1 is structural support and option number 4 is regulation of nuclear pore. The correct option for this question is,
Regulation of nuclear pore.
sorry, can you repeat the answer?
Option A, ma'am.
Option A.
Pores, ma'am.
Okay. So the correct option for this question is question number 4, regulation of nuclear pores. Let's see how this option is correct. So if we see the structure of nucleus and the membrane covering the nucleus is, we have outer membrane and inner membrane which compose the nuclear envelope. So the inner membrane has mainly has function in regulation of the nuclear pore because it interacts with the nuclear pore complexes embedded in the nuclear envelope. So these complexes, they control the selective transport of a molecule, which can be mRNA, protein or other ribosomal subunit between the nucleus and the cytoplasm. So whereas the mRNA synthesis, it happens in the nucleus, but it is not function of inner membrane of mRNA synthesis. And also the the inner membrane can also be involved in the structural support, but the main function provided for the structural support is the outer membrane and the matrix of the nucleus, which is present inside the inner membrane. The main function of the inner membrane is the regulation of the nuclear pore, which is involved in the selective transport of molecule. It is also responsible for the structural support, but it is not its main function. The structural supports mainly come from the matrix, which is connected to the different sides of the membrane and also the outer membrane. So the correct option, if we go just for single option, the correct option is the regulation of nuclear pore. I hope this question is clear. So let's move to our next question, question number 5. The role of nuclear matrix attachment site in chromatin organization. A. Facilitate nucleosome assembly. Second, anchor chromatin to nuclear envelope. 3. Induce chromatin condensation. 4. Regulate histone modification. Please try to answer this question. We have one option, option number 1. We have another option, option number 2. Is there any other option that might be correct apart from option number 1 and 2? So the correct option for this question is option number 2, anchor chromatin to the nuclear envelope. So the chromatin, so basically DNA is present inside the nucleus in a condensed form, chromatin, and further chromatin further condensed in the form of chromosome. So this DNA present as a chromatin in the form of chromatin, they has a nuclear matrix attachment site on them, which are specific DNA sequence that serve as anchoring point. So what these sites are, they connect the chromatin to the nuclear matrix. So which basically connect the chromatin or the chromosome to the nuclear envelope. Why it is important? It provides the structural stability to the organization of chromatin loops, which is essential for higher order chromatin structure. So this attachment of the chromatin to the nuclear envelope, it is essential for chromatin uh, to have its stable form. Since the chromatin is a very condensed form of a DNA, so in order to keep it condensed, so there are certain nuclear matrix attachment site which is responsible for the stability and the structure of the condensed chromatin. So the correct option is option number 2, anchor chromatin to the nuclear envelope. I hope this question is clear. Okay, so let's move to our next question, question number 6. Um, the significance of euchromatin and heterochromatin in terms of gene expression is first, euchromatin is inactive, heterochromatin is active. The second option is euchromatin is less condensed and active, and heterochromatin is condensed. Third option is euchromatin is condensed and heterochromatin is less condensed and highly active. The fourth option is euchromatin and heterochromatin have no impact on gene expression. Please try to answer this question. So we have one option, option number 2. Gene expression basically means the protein synthesis from the DNA, the process of, so like the replication, the transcription and the translation. This is how the gene in the DNA get expressed. So what is the significance of euchromatin and heterochromatin in terms of gene expression? Is there any other option that might be true apart from option number 2? the correct option is for this question is option number 2. Euchromatin is less condensed and active, and heterochromatin is condensed. So basically, we have two form of chromatin. One is euchromatin, and another one is heterochromatin. The difference between the euchromatin and heterochromatin is the condensation of both the form. The euchromatin is very less condensed, and due to this less condensation, we have DNA region which is accessible for our machinery, which is responsible for its replication, transcription, and translation. While in the condensed form, in the heterochromatin, these regions are not accessible for our machinery, which is responsible for its replication. So they are inactive. They do not replicate. While this one, they are transcripted. They do not. They are not transcriptionally active, while this euchromatin, they are transcriptionally active genes, because they are more loosen up, and these DNA region where the transcription has to happen, they are accessible to certain enzymes which are responsible for its transcription. So the correct option would be option number 2. Euchromatin is less condensed and active, while the heterochromatin is condensed. I hope this is clear. This question is clear. So if this question is clear, let's move to our next question. Question number 7. The component of the nuclear envelope is directly involved in the recognition and transport of protein into the nucleus. First, nuclear matrix. Second, outer membrane. Third, nuclear pore. Fourth, inner membrane. Please try to answer this question. We have one option, option number 3. We have another option, option number 2. Is there any other option that might be correct apart from option number 2 and 3? So the correct option for this question is option number 3, nuclear pores. Let's look into the concept for this question. So what are the nuclear pores? So the nuclear pores are the structure in the nuclear envelope which are responsible for the recognition and the transport of protein, and this is very selective transport. Not just protein, mRNA as well as other molecules in the nucleus. They allow the selective import and the export through a process that involves the transport receptor like importin and exportin. So in this selective movement, certain protein receptor are responsible for this selective transport of protein or mRNA or other molecules like importins and exportins. They recognize the signals on proteins such as nuclear localization signal. So there are certain signals which are present in the protein. Only when these protein importin and exportin they recognize those signal, only then they allow the selective transport of protein or the other molecular through the nuclear pore. So while the nuclear matrix it provides the structural support, it does not help in the transport of protein or other molecule. The outer membrane it does not, it is again does not involve in the selective transport. It provide the structural support as well as people are still joining in, I'm not sure, like okay. While the inner membrane it interacts with the nuclear lamina, it is not directly involved in the transport. What involves directly in the selective transport is the nuclear pore. So the correct option for this question would be option number 3, the nuclear pores. I hope this question is clear. Okay, so let's move to our next question. Question number 8. The main driving force for the rotation of ATP synthase rotor during ATP synthesis is first citric acid cycle, second electron transport, third proton gradient, fourth oxygen consumption. So please try to answer this question. We have two option numbers. Okay, so we have option number 3 as an answer in the chat box. Okay, so is there any other option that might be correct apart from option number 3? So the correct option for this question is 3, proton gradient. So during the ATP synthesis, which is also called as oxidative phosphorylation, so ATP synthesis is occur in mitochondria. So the mitochondria also has two membrane, the outer membrane and the inner membrane. and in between these two membrane they have intermembrane space. So before this ATP synthesis, there is another process which is electron transport chain occur, which generates the accumulation of these proton in the intermembrane space of the mitochondria. So while there is more number of proton in the intermembrane space and less number of proton in the matrix of the mitochondria, it generates a proton gradient across these across the across intermembrane and the matrix. So then the proton from intermembrane, it goes inside the matrix due to this gradient difference, and when uh, it goes inside, so it rotates the ATP synthase which present in the inner membrane of the mitochondria, which when it rotates, it changes its subunit, it changes the structure of the subunit, which uh, ultimately help in the synthesis of ATP from ADP and inorganic phosphate. So the main driving force for the rotation of ATP synthase rotor during the ATP synthesis is the protein motive force, which is a concentration gradient of proton across the membranes, causing proton to flow through the ATP synthase complex, driving the rotation of the C ring. Here the C ring present in the inner membrane within the F naught subunit. So when this C rings rotate, uh, it leads to the generation of ATP from ADP and inorganic phosphate. So the correct option is proton gradient. So I hope this question is clear. I hope the question is clear. If the question is clear, we can move to our next question. Question number 9. ATP synthase generate ATP during oxidative phosphorylation by first directly phosphorylates ADP using energy from electron transfer. Second, translocate proton down their concentration gradient. Third, catalyzes the breakdown of glucose. Fourth, facilitate the synthesis of NADH. Please try to answer this question. So we have one option, option number 2. We have another option, option number A, option number 1. Is there any other correct option apart from option number 2 and 1? Guys who are joining late, please try to join early by 6:05 from the next class. Else I'll again have to come back and give you the permission. So please try to join the class early, because it has already been 1 hour since the class.
Excuse me, ma'am.
Yes.
I had a doubt.
Yes.
The first session is from 6 to 8 o'clock, ma'am. The other session is from 7 to 9, ma'am. Which session is started, ma'am?
Sorry, you are not clearly audible. Can you speak a little louder?
The first session is from 6 to 8 o'clock, ma'am. We had the other session from 7 to 9, ma'am.
Yeah, there are two.
Which one?
Yes. So the question would be the same. So based on your timing and your feasibility, you can join any of the session.
Okay, ma'am. Thank you, ma'am.
Yeah. So for the 7 to 9, this is being taken by another TA, and for 6 to 8, this slot is taken by me. So I think the links are different as well. So I am not sure why people are joining this link for the 7 p.m. class. So if students are joining for the 7 p.m., the link is different. So this session has already been started from 6. So for the 7 p.m, there is another link for the meeting. So those who are joining now, you can join the another session, because it's already like 1 hour for this session. We have completed many questions till now. So the correct option for this question is option number second, translocate protons down the concentration gradient. So in the previous question, as I have mentioned, so before the ATP synthesis process, there is one more process, which is electron transport chain, which generate the proton in the intermembrane space of the mitochondria, which due to the difference in the intermembrane space and the matrix, this generate the proton motive force, which is responsible for the translocate of proton down their concentration gradient. So the correct option would be option number 2, translocate proton down their concentration gradient. This basically responsible for the ATP synthase to rotate its C ring, which ultimately lead to the synthesis of ATP. I hope this question is clear. Okay, so let's move to our next question, the question number 10. The final electron acceptor in the electron transport chain is first NAD plus, second FAD, third oxygen, fourth cytochrome C. Please try to answer this question. We have one option number 3, oxygen. Is there any other option that might be correct apart from option number 3 for this question? The correct option for this question is option number 3, oxygen. So okay, so let's begin what is electron transport chain and why it is important. So as I previously mentioned, so before ATP synthesis we need the proton gradient across the intermembrane space and the matrix of the mitochondria. So from where this proton gradient happens, or the generation of the protons occur, it occurs from the electron transport chain. What is electron transport chain? It is a series of protein complex 1, 2, 3, 4, and the other molecules like cofactor quinone and the cytochrome C in the inner mitochondrial membrane in eukaryotes or in the plasma membrane of prokaryote. So it, I have already mentioned, it plays a critical role in the ATP synthesis using the energy from electron transfer through the chain. So the main purpose of electron transport chain is to generate a proton gradient across the mitochondrial membrane, which drive the synthesis of ATP through chemoosmosis. So there are certain protein complex in the inner mitochondrial membrane, the complex 1, complex 2, complex 3 and complex 4, and where a different reaction happens. So in complex 1, where the NADH, it accepts the electron and transfer them to ubiquinone, and in this process it pumps the proton into the intermembrane space of the mitochondria, where the protein complex 2, which is succinate dehydrogenase, it accepts the electron from FADH2 and transfer them to ubiquinone and to cytochrome C, transfer it to ubiquinone, and while the complex 3, which is cytochrome B C complex, it transfer the electron from ubiquinone to cytochrome C while pumping the proton into the intermembrane space. So while this electron transfer is happening, so one proton is coming out. So from complex 1, 2 protons are coming out into the intermembrane space, while from complex 3, 1 proton is coming out from to intermembrane space. And from complex 4, in the complex 4, the transfer of electron from cytochrome C to the oxygen occur. So the oxygen is the final acceptor of the electron, and it also pumps the proton into the intermembrane space. So basically this electron transport chain ends when the oxygen accepts the electron and pumps out the proton. So this entire electron transport process, it involves in the generation of the proton gradient across the membrane, the mitochondrial membrane, which further leads to the synthesis of ATP. When the proton moves from intermembrane space to again the matrix of the mitochondria, it rotates the ATP synthase, and which leads to the synthesis of ATP from ADP and inorganic phosphate. So how this proton gradient is generated, and ultimately it leads to the generation of ATP. So the correct option would be option number 3, oxygen. So I hope this question is clear. So let's move to our next question, question number 11. During the ATP generation in mitochondria by ATP synthase, proton moves from first from the intermembrane space to the matrix, second from matrix to the intermembrane space, third both directions simultaneously, fourth from intermembrane space to the cytoplasm of the cell. Please try to answer this question. So we have one option, option number 1. Is there any other option that is correct from apart from option number 1? So the correct option for this question is option number 1 only, from intermembrane space to matrix. So as I previously mentioned, so during the electron transport chain, the protons are being pumped from the matrix to intermembrane space, which generate the proton motive, which generate the proton gradient, which leads to the generation of proton motive force, from which the proton again gets inside from intermembrane space to the matrix and change the conformation of ATP synthase, which lead to ATP formation. So the proton, they move from intermembrane space to the matrix of the mitochondria. I hope this question is clear. So if this question is, so the correct option is option number 1, from intermembrane space to the matrix. So if the question is clear, let's move to our next question. Question number 12. The membrane within the chloroplast that contains photosynthetic pigments and is the site of the light dependent reaction is first outer membrane, second inner membrane, third thylakoid membrane, fourth stroma. Please try to answer this question. So we have one option, option number C, thylakoid membrane. is there any other option that might be correct apart from option number 3? So the correct option for this question is thylakoid membrane, the option number 3. So this is the structure of chloroplast, and here this is the stacking structure, it is thylakoid. So this thylakoid, the membrane of this thylakoid, they contain the photosynthetic pigments such as chlorophyll, which are crucial for the light dependent reaction. So basically light dependent reaction is the reaction which ultimately leads to the generation of ATP and NADPH. This is again like the reaction which is similar to the electron transport chain that we have seen in the eukaryotic cell in the mitochondria. This is while in plant, this reaction occur in the thylakoid membrane, which leads to the generation of ATP and NADPH in presence of light, which these ATP and NADPH then are utilized by light independent reaction, which is the Calvin cycle that occur in the stroma of the chloroplast, which ultimately lead to the formation of glucose, because plant, they produce their own food. Plants are not dependent on another for their food, they generate their own food. So we have two reactions. One is light dependent reaction, which leads to the generation of ATP and NADPH, and the other is light independent reaction, which uses these ATP and NADPH produced during the light dependent reaction and ultimately synthesize an enzyme which is responsible for the glucose formation in the plants. So this reaction occur in the thylakoid membrane. This light dependent reaction first occur in the thylakoid membrane, and while the light independent reaction, it occur in the stroma of the chloroplast. So the correct option is thylakoid membrane. I hope this question is clear. So let's move to our next question, question number 13. The function of the stroma of chloroplast: first, capturing light energy; second, synthesizing ATP; third, carrying out dark reaction of photosynthesis; fourth is oxygen consumption. So please try to answer this question. We have already discussed in the previous question. We have one option, option number 3, carrying out dark reaction of photosynthesis. is there any other option that might be correct for this question apart from option number 3? The correct option for this question is option number 3, carrying out dark reaction of photosynthesis. So in the previous question, I've explained that the light reaction, it happens in the thylakoid membrane, while the dark reaction happens in the stroma of the chloroplast. So after the light reaction, um, which has generated ATP and NADPH. So in this, the those generated ATP and NADPH are consumed by the dark reaction. So it is the carbon dioxide fixation reaction which utilize the ATP and the NADPH that were previously generated by in the light reaction. So it ultimately lead to the formation of glycerol aldehyde 3 phosphate, which is an enzyme responsible for the glucose, ultimately responsible for the glucose formation. So the precursor is this glycerol aldehyde 3 phosphate, which ultimately lead to formation of the synthesis of sugar and other organic molecules. So this reaction, it happens in the stroma of the chloroplast. So the correct option is carrying out the dark reaction of photosynthesis. So I hope this question is clear. So let's move to the next question, question number 14. The primary function of Golgi apparatus in vesicular trafficking is first synthesizing proteins, second degrading cellular waste, 3 storing cellular energy, 4 modifying and packaging protein. Please try to answer this question. We have one option, option number D. Is there any other option that might be correct apart from option 4? So the correct option for this question is option number 4, modifying and packaging the proteins. So let's look into the concept. So basically the Golgi apparatus, it is primarily responsible for modifying, sorting and packaging of proteins and lipids that are synthesized in the endoplasmic reticulum. So once the protein are synthesized in the endoplasmic reticulum, they are transported to the Golgi apparatus in vesicles. So in Golgi, these protein they undergo further modification such as addition of carbohydrate group, glycosylation, and are sorted and packaged into vesicles for transport to their final destination. So basically, when the protein are first synthesized in the endoplasmic reticulum, they are not functional protein. So to become uh, they has to become biologically active for their main function. So there are certain further modification and packaging of those protein required, so they can finally be functional and perform their biological function. So these modification and packaging and of the protein, this happens in the Golgi apparatus. So then from endoplasmic reticulum, uh, the protein goes to Golgi apparatus, they get modified, and then they either come to the cell because there are certain protein required in the cell, or they get out from the cell and perform their specific function. So the correct option for this question is modifying and packaging of protein. So I hope this question is clear. So if the question is clear, let's move to our next question. Question number 15. The primary function of a laminar flow hood in a cell culture laboratory is first, to increase the CO2 level. Second, to provide a sterile environment. Third option is to regulate temperature and humidity. Fourth option is to maintain proper lighting condition. Please try to answer this question. We have one option. Option number B, to provide a sterile environment. The correct option for this question is option number 2, to provide a sterile environment. So let's go to this link. I'll also provide this link in the chat box so you guys can read more about the laminar hood. So laminar hood are basically you cannot see. So basically they are a working cabinets or enclosed workstation that is used to create a contamination free work environment through filters to capture all particles entering. So these laminar flow, they are provided with HEPA filter, so that an airflow in this laminar is such a way that there is no contamination of any bacteria or any other organism that is present inside this laminar flow. So when we have to work in the laboratory, we have to work in the sterile condition. So we generally work in this cabinet which provide the sterile environment. So you guys can go through this link, and you guys can read about how many kind of laminar flows hood are there, and why there are different kind, and what specificity of each laminar flow hood is there. So you guys can read about it in this link. So the correct option for this question is Question option number 2, to provide a sterile environment. I hope this question is clear. So I hope the today's lecture was clear. That was our last question. If you guys have any doubt in today's lecture, you can ask.
Ma'am, we should submit any assignment.
Sorry.
We should submit any assignment.
That I am not sure about. If the professor has given any assignment, then you should. Apart from that, I don't think so. That I think these are the assignments that you need to solve and submit. If I am not wrong, but once you can confirm in the portal.
Yes, ma'am.
Okay. Because these are the assignment questions only, so I think you guys need to solve these questions as well and submit the assignments. But please do confirm on the portal. Are there any other questions? So if there are no other questions, we can end our session and we will meet again the next week at the same time. Thank you all for joining in the session. So we can end our session here. Thank you for thank you all for joining the session.
Thank you, ma'am.
Thank you, ma'am.